Type 1 diabetes is an autoimmune disease in which insulin-secreting pancreatic beta cells are destroyed by autoreactive T cells. Understanding the mechanisms involved will provide insights to generate an effective cure for this disease. Here, two methods to observe specific CD8-positive T-cell-mediated destruction of pancreatic beta cells in vitro are demonstrated. A chromium-51-based cell-mediated lymphocytotoxicity, or CML assay, and a visual CML assay. For both methods, CD8-positive T effector cells are first prepared from mouse spleens. To perform a CML assay, NIT1 or NIT4 target beta cells are labeled with chromium-51 and co-cultured with effector cells. When target beta cells are killed, the chromium-51 is released. The supernatants and cell lysates are then collected, and radioactivity levels, which indicate the extent of cell death, are measured using a gamma counter. To perform a visual CML assay, NIT1 and NIT4 target beta cells are labeled with mitochondrial potential dye TMRM. Effector cells are labeled with picogreen. The cells are combined and live cell imaging is performed to examine the rate of cell death, indicated by dissipation of mitochondrial membrane potential.
The main advantage of this assay over traditional assays such as the chromium release assay is that we can observe beta cell death in real time. Additional advantages include the ability to determine whether CD8 T cell mediated death of beta cells is contact dependent. And also, this assay can be extended using